Okay, so tonight we're going to talk a little bit about how to protect the brain, and this will apply to the spinal cord as well. Uh, Basically, how to protect it from your skull so that it's not crashing into the walls of your skull that help protect it from outside damage. So, we are going to focus first on cerebrospinal fluid. Okay, and cerebrospinal fluid is going to be abbreviated as CSF. And so cerebrospinal fluid is made from the choroid plexus. And if you'll remember, the choroid plexus is, I see these, I know it's a black and white picture, so it's not ideal. I see these here. This is all choroid plexus. I see choroid plexus back here at the top of the fourth ventricle. We'll talk about that in a little bit. But in that epithalamus, above the thalamus area, you're going to see a lot of this choroid plexus. Choroid plexus is made mostly of ependymal cells. If you remember from our neuroglia, those ependymal cells, their job, right, was to make, move, and monitor that cerebral spinal fluid. And so the choroid plexus is made up of these ependymal cells and blood vessels. So cerebrospinal fluid is going to be very similar to the composition of blood plasma. For those of you that aren't familiar with blood plasma, that's basically the watery part of our blood, not the red blood cells, not the white blood cells, just the watery part of the blood. So the consistency and the makeup of the two is going to be very similar. It's going to be made by that choroid plexus. Okay, and in particular, these ependymal cells, they're really going to help, kind of filter the blood and help produce this cerebral spinal fluid. And it's going to circulate around the brain through the ventricles and the subarachnoid space. And it's going to circulate around the brain. It's going to circulate down through the spinal cord, around the spinal cord, and it's going to help to protect the brain. It's going to help to nourish our central nervous system, and it's going to help cushion it. Remember, this is a fluid, so it's going to provide a fluid barrier here between the brain and the skull itself. So if you think about it, if we put something soft in like a 10-gallon bucket, right? Let's put a basketball in a 10-gallon bucket. If I swing or move that bucket around, the basketball, right? The basketball is going to crash into the sides of the bucket. Okay. That'd be your brain, right? If I take the same 10-gallon bucket and I fill it with water and then I put the basketball in it, it's not necessarily going to crash, right? It's going to kind of float around and kind of and move with the water and swing with the water. And so the water will help protect that basketball from the walls of the bucket. And so that's a little bit of how your CSF will work to help protect you there. Okay, I'm going to pause for a second and clean all this up, and then we're going to label some stuff, and we'll be all set. Okay, so in addition to our cerebral spinal fluid helping to protect the brain, we also have the meninges. The meninges are a covering of the central nervous system tissue that are going to help kind of anchor it in place and wrap around it and help protect it. So we have 3 layers to the meninges. The outermost layer is the Dura Mater. When you guys do your dissection next class, you will see this layer. It's pretty hard. It's actually kind of difficult to cut through. It's very hard. It's very tough. Okay? It's thicker than the other layers. It's actually a double layer situation where you've got one side right up against the skull and then one side on top of the brain. Okay? And that Dura Mater would be right here on the inside of the skull. And in that Dura Mater, in that subdural space, so let me spell some things for you. So in that subdural space between the 2 layers of the Dura Mater, you're going to have the dural sinuses. And again, this is something that you're going to see when you dissect your brain. They'll look kind of black because they will have some leftover drained blood and such in them. Okay? But the dural sinuses are where the cerebral spinal fluid is going to end up draining to. Okay, so hopefully this image here will help a little bit. I kind of on purpose have chosen one where you can't really see the names because what I'm afraid is some people will get bogged down in these names. So as we talk about these dural venous sinuses, you can see them here, right? And there's one across the base, and there's one back here. So these dural venous sinuses are located in what we call the dural folds. Remember, we said that the Dura Mater was a double layer. And so these dural folds dive down into areas. For instance, this right here, this dural fold is in that longitudinal fissure. They dive down into areas where there's basically gaps in the brain tissue so they can help anchor it and, kind of lock it into place here. And so some of these dural folds have names, like for instance, this dural fold right here, all across here, that one is called the Falx cerebri. And again, I'm going to find that one down in the longitudinal fissure, and it's going to be filling that space down to the corpus callosum down at the base. Okay, there's another one right here of one of these dural folds that if we have our brain, our cerebrum sitting here, our cerebellum sits under it right here. So our second dural fold that we're going to talk about here is our tentorium, like a tent, Tentorium Cerebelli. So this one sits like a tent on top of the cerebellum. So in this space, if you look at bend back to the picture that you have in the right here, it sits right here. So the falx cerebri would be coming down here between the two cerebral hemispheres. Tentorium cerebelli fold is a tent over the cerebellum. Okay, and then there's a small fold, and let me clean that up so you can see it better. And there's a small fold right here into the cerebellum, right there. That one is called the falx cerebelli, and that goes between the two halves of the cerebellum, okay, and the cerebellar hemisphere. So there's a right and a left side of your cerebellum, just like there's a right and a left side of your cerebrum. So we have these three dural folds. You'll really be able to see the falx cerebri when you do your dissection. It'll have some dural sinuses in it, and you'll see kind of how they're connected and such. Okay, but they're going to contain these dural sinuses, which are going to help drain this cerebral spinal fluid. Our next layer of our meninges is going to be the arachnoid mater. The arachnoid mater means spider-like. Is going to be our second layer. It won't be as thick. It won't be as tough. In this diagram right here, it's going to be this white right here that goes up into these what look like broccoli trees. Okay, that's all going to be our arachnoid mater. So that arachnoid mater is going to be our second layer. It's going to contain a subarachnoid space. Remember, sub is below. So it's going to contain that subarachnoid space. That subarachnoid space is going to be this area here. So all of this dark gray here between the brain and that arachnoid mater layer, that would be our subarachnoid space. That's going to contain CSF. And it's also going to have these little broccoli trees. They have a name. Those are the arachnoid villa or via. Okay, they basically act like little gateways for the cerebrospinal fluid to get out of the subarachnoid space. Your body's constantly making that cerebrospinal fluid, constantly circulating it, constantly producing and replacing it. Okay, and so it needs to filter out. And like we said, it'll drain into those dural sinuses. So it'll come out through these arachnoid villi into these dural sinuses. And we're going to walk through the flow of cerebrospinal fluid in a minute. Okay, our last layer of meninges is the pia mater. The pia mater actually touches the brain itself. It is right there on the brain itself. Very thin layer, really just an extra coating on top of the brain. When you do your dissection, like I said, again, you'll be able to see the Dura Mater, And you won't really be able to see the Arachnoid Mater because it'll kind of blend in with the Dura Mater and get kind of pulled off. And you'll kind of be able to see the Pia Mater. You'll see how it will look shiny, okay, in places, but the Pia Mater is so thin, you can't really pull it off and make it be and really see it as its whole extra layer. So again, it's a very thin layer of protection that's actually touching the brain tissue itself, touching the central nervous tissue, follows every gyrus, every sulcus, little layer of protection there. Okay, so we've talked about CSF and how it is going to protect the brain by helping offer this cushion. It's going to help nourish the brain. Okay, it's going to, we've mentioned these meninges and how the Dura Mater makes these dural folds that'll contain these dural sinuses. So now let's actually trace the flow of cerebrospinal fluid. And we're going to trace the flow of it while identifying some of the structures that it flows through, and then we'll be all set. So if you can see in this picture here, you'll see these, they almost look like shadows right here, these little guys here. Okay, they look like shadows because they're basically a space. Okay, so we're going to be looking at, that is our, let's see, that's the right side of our brain. So that is a right lateral ventricle. And so a ventricle is going to just be these openings. So it's going to be these openings where the cerebral spinal fluid is either made, kind of circulated through. And so we have a right lateral ventricle, and so I also have a left lateral ventricle. So I have one on each side. So if I was looking at a view from the front, right, I'm looking straight at like a person's head, person's face, right, I would have little lateral ventricles there. They'd actually be going backwards, right? And then there's going to be a third what's called the third ventricle in the middle. So this right here is going to be where we find our third ventricle. If you remember from the previous video, we mentioned that we have a right and a left thalamus. If you think about where we see the thalamus located, and again, I have a right and a left side, okay, that third ventricle sits in between the right and left thalamus. So this is between the right and left thalamus there. And then after that third ventricle, I have this little like diamond shape right here in front of the cerebellum. That's my fourth ventricle. So I think they would all be named 1, 2, 3, and 4, except for the fact that I have a right and a left lateral ventricle. And so those are right and left lateral ventricles, and then we just have third and fourth ventricles. So the cerebral spinal fluid, like we said, is made in the choroid plexus, which is kind of located here, kind of in the top of the third ventricle. So as we're trying to kind of track the flow of the cerebral spinal fluid, okay, we're going to kind of start with the choroid plexus, and we'll just start with that one there. So it's going to go through this choroid plexus. Remember, that's going to be made of all those ependymal cells that are going to help make it, move it, and monitor it. All right, so it's going to create this cerebral spinal fluid. Okay, and you'll notice these choroid plexi are also kind of at the base of these lateral ventricles. So the cerebral spinal fluid is going to go from these right and left lateral ventricles. From those lateral ventricles, it's going to drain down into that third ventricle. From that third ventricle, it's going to go through the cerebral aqueduct. So the cerebral aqueduct is just this canal right here between the third and the fourth ventricle. So that's behind the midbrain area there. So that's my cerebral aqueduct. From that cerebral aqueduct, it will go to the fourth ventricle. And then if you will notice, if you look at your black arrows here, your black arrows in this diagram are kind of following your flow of your cerebral spinal fluid. If you look at your arrows in the diagram, you'll see that you have two options from here. You can go down, straight down through the middle, or it can go out to the edges. right? It can go out. You see it wrapping around here and going on the outside. So from the fourth ventricle, the cerebral spinal fluid is going to have two choices. From the fourth ventricle, it will either keep going down the central canal, or it will, and down through the spinal cord, or it will go out into that subarachnoid space. So it can either go into the central canal. The central canal is going to run down the middle of the spinal cord. You can see the little break there 'cause it's going all the way to the end of your spinal cord. So it can either, and then from there, it will go out into the subarachnoid space. So it can either go straight to the subarachnoid space or down the cerebral or down the central canal first. So, but if it goes down the central canal, it will still end up in that subarachnoid space. From the subarachnoid space, remember this is your subarachnoid space here, right? From that subarachnoid space, it's going to go out through the arachnoid villi, right? Your little broccoli trees. So out through the arachnoid villi. That will send it into those dural sinuses. And those dural sinuses are going to drain back into the venous system of the blood, back into the veins of the blood, right? And take that, drain that cerebral spinal fluid out so that you have room for the new recirculating, new cerebral spinal fluid. So right and left lateral ventricles, third ventricle, cerebral aqueduct, fourth ventricle. From there, can take the central canal route or the subarachnoid space route. Either way, it will end up in that subarachnoid space, then out through the arachnoid villi into those dural sinuses, which drain into the veins, which allow you to keep making that cerebral spinal fluid and you not increasing pressure. That's a really big deal that we don't increase pressure in our head because our brain has nowhere to go, right? It would push up against the skull and the brain would lose that battle against the skull because the brain is a lot softer. So that's where you would result in brain damage. That's why you see in some of your medical shows and stuff like that, where they talk about like drilling a hole in their skull to allow it to drain or to release the pressure. Okay? That's what they're doing. They're removing the hard surface that's not flexible so that the brain has room to swell and expand and not get smushed and damaged. Okay, so you're going to see some of these things on your dissection, and you will see this material on the brain and cranial nerve summative.